Could you please explain in simple terms what's optogenetics?
Optogenetics is a form of wireless communication with nerve cells in the brain, just like a radio broadcast is a form of wireless communication with different households in a city. The role of the radio waves in optogenetics is played by optical signals, by light, and the role of the radio receiver by photoreceptor proteins, light-sensitive proteins, just like the ones we have in our eyes. The light acts on these receivers and causes the neurons that express these receivers to become electrically active. And in this way, it's possible to write new information directly to distributed groups of nerve cells in the intact living brain.
How did you get started in this research area and with what purposes?
I came to neuroscience as an outsider. I had worked in a leading cell biology lab as a postdoc, and there I had seen that in order to understand the biological system, it's absolutely essential to be able to control it. When I started in neuroscience, I felt that there was way too much observation and not enough intervention. And the inability to control the function of the brain impeded our understanding of its fundamental operational principles. So I started to think what kind of method would be required to achieve this level of control. It would be a method that would have some built-in biological specificity to address just some neurons and not others. It should be a method that would ideally be non-invasive, and it should be a method that would be applicable in a intact biological system, in a behaving animal. And so as I went through all of these criteria, one Saturday afternoon it just occurred to me that the expression of photoreceptor proteins could solve all these problems at once. And that's how the idea of optogenetics was born. And in experiments that then took many, many years and the efforts of many, many different people, the whole approach was born.
From the possibilities that your technique offers, which aspects do you find more interesting?
I think there's sort of the interesting fundamental principle that every optogenetics experiment reveals. And that is that our mental lives are the product of intelligent matter. That the physical substrate, the brain, causes our emotions, our thoughts, our behavior. And then there's, of course, all the practical aspects, the ability to pinpoint the neuronal causes of behavior with great precision, the ability to map connections between neurons, the ability to test mechanistic ideas that were the motivation for the initial invention of optogenetics. But what excites me most about these applications is not so much the technology, but actually the beautiful biology that these experiments have often revealed.
In what research lines are you working currently?
We currently work on neuromechanisms that are time-dependent, so-called integrator circuits that accumulate information over time. We study two such circuits. One circuit measures time that we have spent awake and puts us to sleep if we exceed a certain limit. And the other one gradually accumulates the information that we need in order to make informed decisions. Integration of information is a fundamental property of the nervous system, but we have only limited understanding how it actually works. And that's what we are trying to find out.